A quiet revolution is underway in the field of psychiatry, one that doesn't begin in the brain but in the gut. Traditionally, mood disorders like depression and anxiety have been viewed through the lens of neurochemistry, trauma, or cognitive dysfunction. But emerging science is rapidly expanding this model to include the gut microbiome and its intimate dialogue with the nervous system. At the heart of this paradigm shift lies the discovery of specialized gut cells called neuropods, which can communicate directly with the brain via the vagus nerve, relaying microbial and nutritional information in milliseconds. This new form of gut neuron signaling is reshaping how we understand the biological roots of emotional distress, and it may mark the beginning of a new era in psychiatric treatment. Neuropods are a type of enteroendocrine cell embedded in the lining of the small intestine. Unlike other cells in the gut, they form direct synapse-like connections with nerve fibers, allowing them to convert chemical cues into electrical signals that travel almost instantly to the brain. This means that the gut doesn't simply influence the brain through slow hormonal cascades or immune pathways. It can send targeted, neural signals that shape brain activity in real time. These signals can be driven not only by nutrients, but also by microbial metabolites such as short-chain fatty acids, tryptophan derivatives, and neurotransmitter-like compounds produced by the microbiota. In the context of mental health, this is a groundbreaking revelation. Depression and anxiety are increasingly understood as systemic disorders, not just confined to neural circuits. Inflammation, disrupted circadian rhythms, gut dysbiosis, and metabolic dysfunction all play a role. Neuropod-mediated communication may be the mechanism that ties these threads together. When the gut microbiome is balanced and diverse, it produces metabolites that can activate neuropods in a way that promotes calm, emotional resilience, and cognitive flexibility. But when dysbiosis occurs, whether through stress, poor diet, antibiotics, or illness, the signal changes. Neuropods may transmit altered messages that upregulate stress responses, increase inflammation, or disrupt neurotransmitter balance in brain regions involved in mood regulation. There's already growing evidence to support these links. Individuals with depression frequently exhibit lower microbial diversity, reduced populations of beneficial bacteria such as lactobacillus and bifidobacterium, and increased gut permeability, which allows pro-inflammatory molecules like lipopolysaccharide, LPS, to enter the bloodstream. These changes have been correlated with higher levels of circulating cytokines, which in turn affect brain function and mood. More intriguingly, these patients also show altered activity in the vagus nerve, a primary communication highway between the gut and the brain. Reduced vagal tone, a marker of decreased vagus nerve activity, has been associated with higher anxiety, lower emotional regulation, and poorer stress resilience. Animal studies have taken these connections even further. In germ-free mice, which lack a microbiome, anxiety-like behaviors and exaggerated stress responses are commonly observed. When certain probiotic strains are introduced, such as Lactobacillus rhamnosus, these behaviors normalize and GABA receptor expression in the brain shifts toward a more balanced state. Importantly, when the vagus nerve is severed, these benefits disappear, proving that microbial effects on mood are mediated through direct gut-brain neural pathways, not just hormonal diffusion. This makes neuropods and the vagus nerve a crucial interface, one capable of modulating emotional states in ways we're only beginning to understand. The implications for psychiatry are profound. Rather than viewing gut-related symptoms as side effects of mood disorders, we may soon regard them as drivers of emotional dysregulation. This perspective offers new targets for intervention. Restoring gut balance through prebiotics, probiotics, or dietary change may not just improve digestion, it may correct faulty mood signaling at its source. Likewise, treatments that enhance neuropod function or increase vagal tone, such as meditation, breathwork, or even electrical vagus nerve stimulation, could become part of a new therapeutic toolkit for anxiety and depression. What makes this model so promising is its precision and speed. Unlike drugs that may take weeks to alter brain chemistry, neuropod signaling can shift mood-regulating circuits within seconds to minutes. This rapid feedback loop between the gut and brain suggests the potential for interventions that feel more natural, more holistic, and perhaps even more effective, especially for patients who do not respond to traditional medications. We are still in the early stages of understanding how specific microbes, nutrients, and gut cell types influence mental health, but the foundations are already in place. Psychiatry is no longer limited to the skull. The gut has emerged as a dynamic player in emotional regulation, capable of influencing how we think, feel, and respond to the world. As we continue to decode this gut neuron circuitry, we may uncover new, non-invasive ways to treat mental illness, ones that honor the deep interconnection between body and mind. In this new frontier, healing the brain may begin with nourishing the gut. And what was once dismissed as pseudoscience is now gaining credibility as a complex, biologically grounded system of communication, one that could revolutionize how we treat and understand human suffering.